So these kind of species are very important, how to understand those species in the pharmaceutical chemistry. And also we developed the base-induced ring opening reaction. A base-induced ring opening also is very important, especially jet dienes. This is a jet diene is a very interesting species in pharma and medicinal chemistry. And as you know, organic chemistry, you will see classical neighboring group participation. The neighboring group participation, how important in especially reactivity, understanding, selectivity, and all. So we try to explain this phenomena in asymmetric organocatalysis. When you are trying to develop the asymmetric organocatalysis, especially catalyst, substrate, functional group, and all, how those neighboring group participation in to direct the organocatalysis for high asymmetric induction. So we investigating this last few years. And well, and also we try to develop the Jepper cyclization with organophosphine catalysis is another phenomena, especially in organic chemistry. We'll have a phase transition state theory, the pre-transient state, transient state theory. So we want to understand more elaborately this transient state theory. So for that, we try to develop the supramolecular organocatalysis, where we can able to stabilize the pre-transient state before breaking the bonds. Before breaking the bonds and making the bonds, how the cluster. We want to make the cluster like this and stabilize the pre-transient state and try to understand. And we work in total synthesis, and also we developed the click reaction, where last year Nobel Prize in click chemistry. Last year Nobel Prize in click chemistry is the copper mediated chemistry got. So parallelly we developed the reaction without using copper, this click reactions, and where he has a lot of application in medicinal chemistry, pharma. And also recently we are trying to develop this lasone. This is the key molecule in our Gorentak. I think all are Telugu people only, or other language people also there here, madam?
Other people are also there, sir. They are from Bangalore, Chennai, Kerala,
So
Mumbai.
Telugu people call this is of Gorentak, and this North Indians will talk as a Enna, and Tamilians, I don't know what name it will be there for this. This is the molecule, key ingredient in that Gorentak or Enna powder. So last few years we are working to utilize this molecule in medicinal chemistry and pharmaceutical concept to understand, to synthesize the important drug molecules. So these are the concepts we are working in our lab. And here I may go very glimpses of information to tell you, but whoever interested, I would request you to go through our website. Is everything information is there on the web, and papers, links, and everything. I would request you to go through this website to see more information on this cutting edge aspects of organocatalysis, click reactions, and pharma and all. Well, before the last year, 2021 Nobel Prize in asymmetric organocatalysis. So this area I am working from the last 20 years. I'm very happy. I am the first generator of this one. So is a Benjamin List and myself, we both worked in the same lab, Professor Carla's laboratory. And he went to the Germany and I came to the India to start independent career. So he is the Macmillan from the USA, and these are the front runners of the organocatalysis. Along with them, Professor Carlos Barba is also supposed to get the Nobel Prize, but the nature is different. At a very young age, he got the cancer, and he went to very early Sorgalok. So that's why he not joined in this club to get the Nobel Prize. But he is the one of big pioneer in to initiate this organocatalysis and to make the great success to develop this concept. Vinay Kumar garu. So the last year Nobel Prize, this is a 2022 is a chemistry. This is the catalytic click reactions. He is the Professor Meldal, is a Sharpless and Petrozzi. And Meldal and Sharpless simultaneously they developed this reaction, reaction looking so small, no? How simple? You take the alkyne and take the azide and just add the copper salt, copper iodide, copper acetate, and just ligand and room temperature, little bit heating. You will get the single regio isomer, 1,4 disubstituted. And she understand the importance of this reaction, and she came the strain cyclooctane and added without anything. At room temperature, you can go with there's no zero selectivity in this, but chemistry goes very quickly. And this reaction she performed in the inside the cell, and try to show how important this reaction as a bioorthogonal to see the when we have a camera outside, you can take the picture, right? Different nature, we can take the picture. So this R, if you put the chromophore, and you can go inside the taking the photograph in the inside the body cell and all. So because of importance of this, and this one is a lot of applications in the drug discovery. So because of these applications in pharma, so this reaction got the last year Nobel Prize. Well, so when you all work in pharmaceutical concept and understanding about the one. So here I try to talk about the how to understand to take this important these molecules in one pharmaceutical chemistry. This is a major ingredient of enna or gorintaku plant. These are the molecule, and this molecule have a lot of natural products and antibiotic nature, lot of applications. So you can see the down. These are the molecules in the market in the drug. So we have taken this compound and we used our reaction, reductive alkylation reaction, using amino acid catalyst and different aldehydes and Angers ester, and is Angers ester, and able to go very quick manner, a high yield for to introduce alkyl group there. Can introduce, see that, alkyl group. So we able to make a lot of library of these molecules. Once this library in the hand, then we try to. I hope you people might have seen the Robinson annulation reaction. You may be teaching in organic chemistry. Robinson annulation. In Robinson annulation reaction, normally will be there this ketone won't be there. If this ketone is not there, then if you have 1,3-diketone, then you do introduce alkyl group, then you have methyl ketone, and that will go Michael addition followed by cyclization will go. So if like this, so from here, this carbonyl react will form this kind of species. That is the Robinson annulation. But my interest is when we go Robinson annulation reaction, this one is a nucleophile, this is electrophile. If you go and react with this, intermediates will be forming like that. This double bond will move inside, in electrons will be like that, the OH will form. Then what about this double bond? Whether this double bond is really forming or not, up to now we don't know. Because this carbonyl is there, and this enol will go to keto form, then another enol. This is called this is called endoenol. This is the endoenol, and this will go cyclization. But what about the exo OH? This exo OH, whether it can able to involve in the reaction. To understand this one, because you have one more carbonyl here. So this carbonyl is there. Now here from this is the nucleophile 1, 2, 3, 4, 5. So 1,5 cyclopentanylation is the thermodynamically, kinetically very feasible for the subject to involve in the reaction. So that is keeping in the mind, if this goes and you will get this product. If you react with this and this, these are the products will form. If not going involving here, not trapping, and this goes to endo type, and this goes react with these two, means this kind of product, or if go and react with this carbonyl, you'll get this kind of product. So from this reaction, 6 type of products can form. So keeping this in the mind, we are very curious to understand this reaction. We have taken this substrate, we add methyl ketone and catalyst like 2J, 2K, and 2L. We got exclusively only one product. You see that? Here, this product is this. This product formed this one. This one, how it be going to come? If this is the first formed enolate, will be nucleophile, will go and react with this carbonyl to make cyclopentanylation. So that's what happening here. You see that more than 90% yield, 61:1 DR. This is the diastereomer ratio, and 95% enantiomeric excess, single enantiomer. So almost single enantiomer and single DR. With this, you can understand. You can see how with the 2K, with this catalyst, almost 99:1 DR and 97% E. So after this understanding, so I'm not going to tell you here that we understand with a lot of molecules understanding and finding the reaction. And here is the one we need to explain the reaction mechanism with the control experiments, a lot of control, because I'm not going to talk here all. So whoever interested, then you can see the paper. So with all of control experiment, I try to explain how this catalyst, you see in this no metal, so this is the thiourea component, this is the amine, tertiary amine component. Tertiary amine component will take the H to form hydrogen bonding, and this H will have hydrogen bonding with methyl ketone to induce the polarization, and thiourea component having hydrogen bonding with lactone carbonyl. And this cluster, this here 1, 2, 3 hydrogen bondings here. This cluster will bring the two substrate very close proximity and will involve in the asymmetric reaction to make this kind of molecules, where these molecules are key fragment in many antibiotics. So we are working on to synthesize one of the antibiotic to apply this one in total synthesis. So you can see that how important this phenomena. So here to make this important skeleton, we didn't involve any much. Even graduate students can perform this reaction. These are very simple, very simplified one. Just mix A plus B in the oil, add just catalyst, you will get the high enantiomer selectivity. Imagine if you go some 10-15 years back to synthesize this one in asymmetric version, you need to have an extraordinary planning and all with the argon, metal, ligands, and temperature controlling, going up and down. There's a lot of efforts you need. But the way technology improving, the technology way it is going on, very simplified. That is the way the future. So future when it is coming, that's what, when you use the previously some 20-30 years back, when you see your laptops, their Dell, whatever the computer is heavy, right? Now everything in your small cell phone, you can do a lot of things. So organic chemistry also evolving like that. So more simplification to make the drug molecules very easy manner. Well, this is the we explain with pre-transition state to explain why this is the way major enantiomer and this way goes is the minor enantiomer. What is the energy difference between these two? Because this is the most feasible path and this is little high energy path. Because of this high energy path, it is not involving to make the minor enantiomer. Well, so that's what when we see this importance of this alkyl lactone, we try to apply in other reaction where you can say if this is the TMS OTF, you can make the benzene. When you make the benzene in situ, benzene is a very good electrophile. It can also become nucleophile. It's an ambient molecule, and this is a nucleophile, and this will become electrophile. This is a nucleophile. That benzene will become electrophile. Now anion is there. This anion, how it will go and react with this carbonyl. You see that 3 electrophiles are there. This carbonyl, this carbonyl, this carbonyl. If 3 electrophiles are there, how it will react? This is a question mark. If this fellow goes and react with the adjacent one, because it's highly reactive, right? Aggressive. So don't have time to think any other possible ways. So immediately this will go and react with this carbonyl and will form this kind of alkoxide. If this alkoxide electrons are giving back and this bond is migrating to this carbonyl. Very interesting. This bond migration is taking place, not bond elimination. If bond elimination means you can go just eliminate like that, but not going. Bond with a vectorial mode is a stereoelectronic manner. In the stereoelectronic manner, this bond is migrating to this carbonyl. That's the reason these interesting compounds are formed. These are very interesting compounds. We have examined this reaction 36 examples, and you can see these applications. You see, these are the very important antioxidant and natural product and material oriented important. So we are working on to make one of this molecule, apply this methodology to make this molecule synthesis, total synthesis. Actually, we are very fashionable in organic chemistry where we want to do more than 10 reactions in one pot, more than 10 reactions in one pot to make the drug or natural products with one single filtration. Previously, we have to go step by step, lot of steps and lot of time, effort, money, lot waste. But here, we can have a just mix, take this starting material, mix on 1, 2, 3, 4, like that. So we can make is kind of natural product type molecules where you can go and investigate biological properties. You see here, olefination, transfer hydrogenation, olefination, orbitic reaction, Michael reaction, all 5 reactions are taking place. The 5 reactions went on to make this kind of molecule. If you can see this kind of molecule, how many are there antibiotics and natural products in the right side. So that's the way we develop this phenomena, and we entire phenomena we understand clearly, and we are now present. We are developing asymmetric version of this reaction. Asymmetric version of this reaction we are developing. Well, when we are developing this asymmetric reaction, so we come across, if you take the substrate like this aldehyde, you add the Wittig phosphorane, then olefin will form. You have to add, here you see, very interesting here. If this is the substrate and this is the phosphorane, you didn't add any catalyst, then product form. Product form very nicely, 99:1 DR, and within 1 hour you're able to get the compound very nice manner, aliphatic. You see, when you mix A plus B without a catalyst itself, within 1 hour, the product is formed. Now, you want to add the catalyst, 10 mole % of catalyst between these. You see that 2 hours, 80% yield, 1:1.5 DR is formed, 87% E got. I hope you teachers are following this reaction. Between A plus B, without catalyst, within 1 hour, product is formed. Blank reaction is going on. If you add the catalyst to that one, the reaction took 2 hours, same 80% yield. Instead of single DR, become 1:1.5 DR, and major got 87% EE, minor got 52% EE. How it possible? That is very interesting, right? So keeping this in the mind, we developed this concept. Instead of 10 mole %, we add 30 mole % of catalyst loading. We added 30 mole % of catalyst loading at 25 degrees centigrade. We able to get the expected product almost 94% yield, single enantiomer, more than 20 stone DR. And because we have taken 30 mole % of catalyst, so we able to recover catalyst. We recover the catalyst more than 50%. So that is the very interesting. You can see these are applications of this methodology, and we explain the we explain this transition state, how this catalyst is controlling this reaction, whether to go in this activation mode, whether to go in is opposite diastereomer. Is opposite diastereomer how it is forming, other enantiomer how it is forming. So that's the way we explain this reaction. So when we are developing this reaction, because we have to use 30 mole % of catalyst loading, right? So always, every time is the improvement should be there. One of my referee many times referees will comment, when compared to enzyme catalysis or metal catalysis, there will take the small loading, less than 1 mole %, and less than 1 mole % you can go enzyme catalysis and metal catalysis because they are highly reactive. But when come to organocatalysis, loading you have some problem. Loading we need to go a little bit more loading. So keeping that we developed the parts per million level catalytic asymmetric annulations. So when I see the literature, how many are there this in organocatalysis parts per million level loading? Uh, this is the reaction where is the resolution has taken place. You take the racemic compound like that, you load these kind of bienal based catalyst in 1 ppm. Is just 1 ppm, 1 parts per million. And one resolution has taken place very nicely, 33% conversion. But this is a resolution reaction. It's not a 100%. So Benjamin List developed the Mukiyama-Aldal type reaction. Mukiyama-Aldal, this is the ocylyl enolates and ketone using 500 ppm of this list of Bronsted assay. He has taken at minus 20 degrees centigrade 12 hours, able to get the 97% 99% yield of the Mukiyama-Aldal in 2018. But here minus 20 degrees centigrade needed control. And we understand the importance of this lactone, alkyl lactone. Then we try to develop the first reaction with the nitrostyrenes, because nitrostyrenes are very good electrophiles. If go and beta alkyl nitrostyrene, if it goes like this, or alpha alkyl nitrostyrene, if it goes like that, whether it goes simple or complex structure. We try to understand the subject with the beta phenyl nitrostyrene. No reaction. Very interesting. So to understand this one, maybe because of this beta position, maybe chemistry not going. So we remove the phenyl group, simple nitroethylene. It's very difficult actually. These nitroethylenes are not easy. These nitroethylenes are not easy to prepare and become highly reactive and hygroscopic. So we prepared in the toluene solvent 1 molar solution. Then we added, chemistry went very fast and able to get the two products. Okay, chemistry went to the two products. And we tried to develop this reaction racemic version. Then after that, we went on to investigate the chiral version. And the chiral version, we screened a lot of catalysts. When we are screening 5 mole % chemistry, then we have an idea that the subject can go a bit rate fast, because this is reactive and this is the reactive. Then we went on to decreasing the loading of the catalyst loading. Then you see that 5, 2.5, 1, 1, 1, 0.5, 0.5, 0.4. Even 0.4 loading also within 1 hour chemistry went very nicely. Then we went on to investigate this reaction, low loading, 500 ppm, 0.05 mole % chemistry went very nicely, 12 hours took at room temperature. 0 is first addition, just addition at 0, then followed by at room temperature, 12 hours took almost 82% yield, and 99, single enantiomer. So we are very happy that this is a complex. It's going first to Michael reaction and followed by Henry reaction. So Michael Henry, to make this kind of antibiotic structures, we prepared using low loading. Very interesting. We understand this chemistry with the different functional group and different substrate and different alkyl groups, and we try to understand the gram scale reaction. Chemistry went very nicely, and we try to understand the synthetic applications of this material to understand this one, because this is a very sensitive actually. Here I would like to tell you one thing. Very not easy to characterize this. Characterization, understanding itself took some few months here. When this compound, if you get the crude compound, when you take the DR will be like this, almost more than 16:1. If you do the column, you will get almost 2:1, 1:1. So silica will do epimerization. If you put in the HPLC, it can decompose. HPLC also will create problem. Again, you will get the mixture there. You start with single, you will get the mixture. So what's happening here? See, this is the what happening. Here is the electron withdrawing group, NO2 group, and this is the pulling nature, and this is the electron donating one, and this bond will be breaking very easily. So this will give electron and this will break. So will become a retro Henry. When retro entry goes again based on the acid-base nature of environment, again you can back the entry reaction. So this kind of acid or base induced retro entry and base or acid induced entry is going on. That is the reason your DR is changing. This reaction atmosphere and reaction condition is doing this reaction without catalyst. That is the reason your DR is going down and product is decomposing. So to prevent this one, we have done the tertiary hydroxy protection with a little bit more equivalent of acetate chloride with 1 drop of H2SO4 in room temperature. We protect the tertiary hydroxy OH. Once you protect, then no problem. Then this compound is very rigid and very strong and stable and won't decompose. So during this process of acid chemistry, single enantiomer little decrease 96.6%. So that is a little drawback. Where this is the better than completely spoiling the compound. So we try to explain the reaction mechanism very interestingly. Very here is a very interesting question is nitrostyrene, nitro group is very systematic to go hydrogen bonding with the thiourea. But here nitro one is not involving hydrogen bonding thiourea, but your less one carbonyl is involving hydrogen bonding with thiourea. This is the miracle. We explain, we understand everything through the crystallography analysis. Well, so in continuation, we want to understand this importance of the last zones and all. Whatever I'm showing here is all are the drug molecule in the market. These all are very important molecules. It is in the pharma market. So when you see the literature, how prepare those molecules is the tedious job. A lot of metal, a lot of harsh conditions, and people used to use, and also not that much great library. So then we try to understand why don't we develop the protocol is a simple one where you can use the different people in not to go with the costly metals. So here we try to take using our TCRA reaction. Then you add a KMNO4 sodium hydroxide. This is called Hooker oxidation. Hooker developed this reaction in 1950s, but not much understand. It's not much developed. So here we use this reaction. So this is the reaction. So we can get this one from our reactive coupling reaction. Then you add KMNO4 sodium hydroxide. KMNO4 will do oxidation of the double bond. You got carbonyl and acid. And carbonyl with presence of sodium hydroxide enolization will take place. Then enolization will go to aldol reaction with this carbonyl, will form this aldol reaction. After aldol reaction will go a very nice decarboxylation. We'll get this and followed by oxidation. The oxidative decarboxylation, you can say that word. Oxidative decarboxylation will give the product. You see how you started? Here you started with CH2 aryl will become a direct aryl. So one of the CH2 went on as a carbon dioxide. So here no steering, high yields, high selectivity, high sustainable, easily available starting material, export in many applications. These are the applications. So this is the way explained. And we up to now people don't know about this regio selectivity of this oxidation. So we proved completely taking the starting metal X-ray and product X-ray, starting metal X-ray, product X-ray. We completely proved how important this to prove this concept. And no, recently we try to, as I told you, that we want to develop the one part total synthesis of the natural product. These all are natural products, important. And we try to take this kind of quinones and phosphorane and remain catalysis, these kind of products can form. But we able to see exclusively in quinine catalyst. In quinine catalyst, exclusively we able to get the only one type of product. We able to give single DR and very good yield. Presently, we are working on the enantiomer selectivity of this reaction. See, the very interesting that if you take this molecule, if you want to synthesize this one, you can check yourself how many steps has to involve. But thanks to organocatalysis, that we able to get the very single product, and further we are working on it. we able to make the highlight this reaction in the general cover picture. The front cover shows that fungi, the multi-catalytic one-pot method, also stands as a unique kingdom in the field of synthetic organic chemistry. We correlated our organocatalytic multi-component approaches with fungi. Fungi that will develop very interesting scaffold, can synthesize a lot of molecules, organic molecules, very easy manner. So our organocatalysis also can make the natural products very quick manner. That's what we try to correlate. Our Indian chemist, the Swaminathan, is a professor from University of Madras, one of the pioneer in organic chemistry from our country. So this is the molecule called as a Swaminathan ketone. This is the Swaminathan ketone. So normally, here is this is called Wheelan-Mishra ketone. Many pharmacologists, drug, medicinal chemists all use. You take this kind of compound and take base proline catalysis. This is the what first developed in 1907. This is the 1950 racemic one and 2003 mechanism understood. And this is the azasparagus ketone. This is the 1971. This is the 1971. This is the one started proline chemistry, proline catalysis. So azasparagus ketone, Wheelan-Mishra, these are the two ketones are starting for the asymmetric organocatalysis. That time didn't understand the mechanism. 2003 only understood this mechanism, how it is going, proline catalyzing this reaction. From there onwards, this field evolved as a bigger level. Actually, before this azasparagus and Wheelan-Mishra, our Swaminathan from 1966 from India published the understanding about this kind of ketone. This is a Swaminathan ketone. You see, that is the, but, this is not that much developed. Somehow drawback is, after this 1962, whatever 1971, azasparagus, Wheelan-Mishra, those are evolved bigger level. But Swaminathan ketone not evolved that much because when you use the proline in solvent, it did not form that much. Product did not form at room temperature. If heavy heating only later is formed. So that is the drawback that not developed much this reaction. But applications are plenty. So applications are plenty to make almost more than 450 natural alkalides are there. So these alkalides can get from this, and up to now no catalytic asymmetric methodology, no reports, and high demand. And that's the way when you see in the literature, you see this substrate you take prolinamide 1 equivalent, not a catalytic. You take 1 equivalent, then able to get 70% E. Now went on to make this natural product. Same way, if you take this substrate, racemic proline 1 equivalent, you made the racemic Swaminathan ketone, went on to make the natural product. This is another substrate, again racemic proline, then again 1 equivalent, and went on to make the. See that how drawback that still this many years still not understood this phenomena, how to make, and that is the way we try to work on it to understand this problem. What's the problem? Where it is happening? And we try to correlate the reaction, transfer hydrogenation, and we try to understand this reaction, developing first reactive alkylation reaction. We try to develop the phenomena, and we working on it. We are working on it to make this phenomena how important and to make these molecules, and we are working on to asymmetric version of this reaction. Yeah, with this I will stop here. So thank you very much, madam. So we can interact. The teachers all are there here.